Happy.
That's
right.
cool. Thank you.
I think that's the official go. Okay. So today I'm going to be talking about how we've been implementing some innovative biotransformation and distribution capabilities to really help accelerate that early and preclinical drug discovery space. So I'm going to three topics today. One, I'm going to go over the group. I'm going to go over Merck's Biotransformation and Distribution Group briefly, and then I'll get into the meat of the talk. Where I'm discussing how we implemented a quantitative and qualitative metabolic stability and metabolism assay to really help accelerate chemical design early in programs. And then finally, I'll be wrapping up with ways where we're obtaining rapid tissue distribution information for drugs as well as metabolites, again, in that early drug discovery space. As you can tell, that's where I'm really focusing this talk. So using mass spectrometry-based imaging. So first, what does the Biotransformation and Distribution Group do? We're really embedded in the world of ADME and also within the sub-department of ADME, which is in the PPDM Department. So real quick, let me just go over ADME to make sure we're all on the same page, right? When we take a medication or a drug, whether it's orally or IV, we need to know how it's absorbed, how it gets into the body. And then once it's in, where does it go? We need to know where it's going. Is it getting to the target where your medicine needs to be to do its job? And then how is it eliminated, right? How is it metabolized, broken down? And then how is it excreted? So that's the ADME. So what the group does is really helps understand how drug candidates are metabolized. So that's the biotransformation side, both in vitro and in vivo, to really help inform on these clearance mechanisms. We work a lot with our safety colleagues within Merck to look for potential toxic bioactivation and active metabolites. And we also really help with identifying the potential human dose predictions. And then we do a lot of work with regulatory filings. And then that's on the metabolism side. And on the distribution side, we also help understand how the drugs and the metabolites are distributed to the various tissues, right? Did they get where they need to go? And if so, we really also like to know how much is there. So that really helps us inform, again, access to that site of action, is the tissue retention, accumulation, and again, potential toxicities. And again, always working to get into the clinic and helping with the human dosimetry calculations. So people within the group and people within my group and the scientists are really helped positioned to contribute towards what Merck calls better molecules faster. So Merck has a really big initiatives of how can we design really good chemical matter and drug-like matter earlier and faster and eliminate the bad ones. And I'll discuss how we're really set up to help with that. Merck has moved to a very modality independent, and I'm sure many of you across the industry have seen that, where you have small molecules, peptides, oligonucleotides, fusion proteins, ADCs, you name it. I'm sure it's coming through your pipeline. So we really make sure we can have impact within all those spaces. So the way the group is currently set up is we have subject matter experts in different areas. So small molecule metabolite identification, biotransformation of therapeutic peptides, proteins, and oligos, and also on the distribution end where we do quantitative whole body autoradiography, mass spec, and confocal imaging. And then you'll notice I have the circles combined because we have a lot of people within the group that are cross-trained to do all three, two if not all three of these different areas within the group. And it really helps the flexibility and development of the people, and it also really helps move programs down the pipeline. So one last slide on this, on kind of the way we're set up. Again, our jobs really help the advancement of drugs to and through the clinic, and that always progresses with support of metabolite identification. Again, Merck is modality agnostic, and that doesn't mean all modalities, it means the correct modality based on your target and location and so forth. So again, primarily small molecules, peptides, proteins, and oligos. We do all the in vitro and in vivo metabolite identification across MRL, so across the pipeline from early discovery all the way through phase 2. We do things like metabolic soft spot optimization, ID of, again, potential reactive metabolites. We support our preclinical candidate selections. We help build the IVIVCs for safety. And again, we're responsible for the clinical programs as well. So the phase 1 first in human and metabolites and safety testing, as well the phase 2 human ADME studies. As Joe can attest to, we have a whole range of instruments within the group. We have a lot of fun toys. So we have SciEx instruments to do swath. We have multiple ion mobility instruments from multiple vendors now. We have multiple Orbitrap instrumentations. So again, if you're going to have a modality agnostic pipeline, you have to have a lot of tools to cover what you need to analyze properly. And then finally, and this always seems to be the biggest barrier, is data analysis solutions. And we put a lot of effort into identifying the right software. And even in some cases, we've developed our own software. All right, so with that introduction, I'm going to get into the two major topics. And the first one is implementing a quantitative and qualitative metabolic stability and metabolism assay to really help accelerate chemical design, and primarily within the early space. And I really want to acknowledge and give credit to the project lead on this, Dr. Christopher Kachansky, to really help spearhead this project. And actually really has driven it to production. All right, so let's go over the design make test cycle and how we build partnerships throughout MRL, or Merck Research Laboratories, where in the design phase, the chemists help, as along with other scientists in the team, help design molecules. They're made primarily through medicinal chemistry, and then we test them. We test them for metabolic stability, and in this speaker's case here, metabolic instability. And then that information gets fed back, and it's really a collaborative process where whether they're from my group or within the ADME department, we really work closely with medicinal chemists to come up with the next proper design cycle, and then these cycles go over and over. And it's not just, and I'm sure, again, many of you have had to deal with this. It's just, you can't just do this alone, even within an organization such as Merck, and even with their resources. You really need to go outside and sometimes work with outside partners. So I'm going to be discussing a collaboration we put together to build this assay with the CRO, Q2 Solutions, as well as software solutions from Lead Molecular Design and Molecular Discoveries, and in some of the cases, Waters and Thermo and other instrument vendors. All right, so let's step back a little bit. Let's turn the clock back 5 years on the metabolic stability assay experiments that we're conducting within Merck. So we used to do, or sorry, we still do our incubations at 0.3 micromolar. So small molecule incubations here, and various species, in vitro incubations, set time, temperature, multiple time points. And then these were all run on a triple quad, whether that was in-house or out at a CRO. And then all that data was processed to get the intrinsic, quantitative intrinsic clearance values from the triple quad data using multi-quant, I believe. And then that went into our Merck database, where teams could then see the data using ChemCard or Spotfire. And then when the teams wanted to do MetID, they would review this data, cherry pick, and then the process would start all over at a higher concentration. So now we're up to 5 micromolar, and we would repeat the same exact incubation, at least in terms of time points, and then now run it on an HRMS system. So back in the day, we were doing it on a Waters QTOF, and then that data was processed by Metabolinks, and the report out was a PowerPoint. And I'm wondering if many of you have to deal with this. Data was going into PowerPoint files and slide decks. And then these slide decks were put into various folders and team sites and your electronic notebooks. And unfortunately, data was honestly residing in PowerPoints. It was just a big headache. And the other thing of this entire process was taking over a month, because you're going through 2 cycles here. So about 2 weeks for the intrinsic clearance and another 2 weeks for your qualitative MetID. So we started making, over the past 5 years, we've made some pretty fundamental changes. And the first one was to centralize the metabolite identification activities all to West Point. So it's now a centralized group, the Biotransformation and Distribution Group, which is located in the Pennsylvania site for Merck. We've strictly gone to UPLC. I know that may sound funny to you, but there we had a multitude of chromatography instrumentation back in the day, but now we just use UPLC, and it's really helped to provide chromatographic speed, resolution, and sensitivity, as you know. We've expanded our toolbox, which I'll discuss, to multiple vendors. I really don't want to be handcuffed to any one instrument. I think all the instrument vendors make incredible hardware. I think all the instrument vendors make really good instruments that have advantages. So I would like to make sure I could use those instruments and not be tied into one platform because of software. We really wanted to make sure, get away from PowerPoint files, and we made sure we had a searchable storage for drug metabolism data, or drug metabolism database. And so with that, we found, and we utilized mass metasite and web metabase from Molecular Discoveries. And that's really helped open the door to being instrument agnostic. And let me discuss that here. So now, as I mentioned, we really expanded our mass spectrometry toolbox to include multiple vendors and different mass analyzers, and also different acquisition modes. So we use both data dependent and data independent type data acquisitions. So we have multiple Waters instruments. So we have a Vion and multiple Q-tops. So we can do ion mobility and HT-MSE in a data independent acquisition fashion. We have a Q-top, a Sykes Q-top 6600, so we can do swath. We have multiple thermos, and our latest and greatest, we just got a Bruker timsTOF Flex. So now we can do even passive DBA type acquisitions. But this leads into another problem, right? Each vendor has its own acquisition and metabolite identification software. And you really cannot expect everyone in your group to become subject matter expertise in all these different software platforms. It really drives down efficiency. It sounds very, don't want to be a production line, but it just really causes headaches. And people would actually not use certain instruments because they didn't understand how to properly analyze their software. So that's really a no-go. So what we've done now is we now have mass spec agnostic data processing. So we're using Molecular Discoveries mass metasite, which is able to process and analyze all the different vendors' raw files, so to speak, and process them. And then we can now use that as an agnostic platform for looking at our data. It's really efficient. So then the next step is putting it into a database. So Web Metabase is the name for the metabolite database for Molecular Discoveries. So after the data is processed, it goes into this bin, so to speak, in the server for pending experiments, where all the scientists within my group, and myself included, can actually, we review the data and we approve the metabolites. So it's not all completely automated. There's still a scientist who reviews the data. And it's all web-based, which is also really, really helpful. So you can, and I'll show you, you can even do this at home. So it's been really, really helpful during the pandemic and the COVID restrictions, where we can have 1 or 2 people in the lab and most of us at home who are still able to chug out the data and reports. And then those go into a different area in the server for approved experiments. And then that goes out to our customers. And the approved experiments are locked. So people, other teams and our customers can look and have access to the reports and the data, but they can't change anything. So it's locked down. It's really nice. All right. So let's go back. So before the metabolic stability quantitation of the intrinsic clearance calculations, and then the separate qualitative assay, or the MET-ID, was about a 1-month process. So this test one, which was the intrinsic clearance, was historically run in a linear fashion where the intrinsic clearance was really informing on what selections to make for MetID, and then this was done again. And as I mentioned, it took about a month. So now with all the new HRMS systems and software, can we just combine everything? And as you know, that's what we've done. We're just going to combine now everything into one run. And instead of running it on triple quad, run our incubations on an HRMS system to collect all the data. So you can see your loss of parent and calculate your intrinsic clearance. And at the same time, you're doing full scan and data independent type data acquisitions or even data dependent. And you're collecting both sets of data at the same time. So we'll be able to get and focus on, in some cases, more of a soft spot, but I'll get into that for metabolite identification. All right, so let's discuss how you try to implement a new assay at a very large company. And I'm sure many of you had to deal with this also. You can talk the talk and show the papers, but everyone wants to see the data. And everyone was very clear to us that how important the intrinsic clearance data was, so don't mess it up. They didn't care if we added metabolism data to our data packages, but we had to really make sure we didn't mess up the intrinsic clearance calculations. So we had to, even though it's in the literature and many people have done it, we had to show that you can calculate and get the same values of an intrinsic clearance for metabolic stability on a triple quad, which is here on the x-axis, and compare that to the same exact incubations being run on two instruments, just on different HRMS systems, and get a linear correlation here, which we did. And so you can see we did it on multiple instruments. I'm sorry, it's a little small. 6600 in a data-dependent fashion. On a 6600, you would be using swath, and also on a Thermo QE Plus using data-dependent acquisition, and you get the same data across different instruments. So it's a really nice test. And then the next thing, if you're going to move this outside to a CRO, you need to make sure you can reproduce it at the CRO. So this is the data from the intrinsic from the in-house, and this is from the CRO. And again, you can see within twofold error, we're getting most of the same values. So we're able to transfer the assay out to a CRO. And then the next question was, can we successfully do MetID at 0.3 micromolar, where historically we ran everything at 5 for the qualitative assay, if you can remember. So what this graph is showing was a couple of things that we wanted to show. Can we get a metabolite identification of greater than 1 metabolite for 50% of the compounds tested in total? And here what this graph is saying, did we identify a metabolite? Yes or no? This is the total number of compounds in this test set. And on the y-axis here, you have the intrinsic clearance. So as you can see, as compounds are turned over faster or are more unstable, you're going to see more metabolites. That makes sense. And for the few that we didn't see any metabolites, you can see they're pretty stable in microsome. So again, that makes sense. And then can we ID greater than 1 metabolite, greater than for 75 for the compounds? And that's what this line is showing. And so the answer was yes. So when you have a higher clearance, here there's arbitrary cutoff at 100 microliters per min per mg, we definitely see metabolites. So that was really nice to see. And again, let us move that forward. And then we also implemented some automatic MetID data processing and filtering. So we needed to optimize the mass metasite settings, and that was taken care of. All right, so let's go over how the assay is today. So compounds are picked and sent to a CRO where they do the incubations at Q2 solutions, again at 0.3 micromolar as we discussed. And another thing, well, let me back up for a second. The other thing I didn't realize was not all CROs have HRMS type instruments. They're with a basely triple quad. Mostly they have triple quads, and very few had HRMS systems. So Q2 solutions had a QE plus And so then we put the assay on a QE+. But to do that, they don't have a independent data acquisition, they only have data dependent. So they need an inclusion list. So in-house, we find out what the compounds are, and we build the inclusion list of theoretical metabolites, and then they build the sequences at Q2 Solutions, and then they run the assay. So they're doing both following the parent and loss of parent and identifying the potential metabolites. And then we've used GUBS software now to calculate the intrinsic clearance. So that's done at Q2 Solutions. But then the qualitative information actually gets packaged and sent back to us because we have mass metacyte on our servers. And so that's all processed in-house. And then that goes in, again, using molecular discoveries and mass metacyte. And then that all gets put into the Merck database. And that's how it's shared with the teams. And so you'll notice now there's even a link for each compound. So that's what these little blue highlights are. But then the nice thing is now the entire process takes less than 2 weeks. So this is the way we now do our really like a tier 1 quantitative, qualitative, metabolic stability, and MetID assay. So real quick, let me review kind of what the data looks like and some of the data quality. So again, this is that little link. If you click on the link, it'll open up with this browser. And this is kind of what the reporting structure looks like for our teams. So this is verapamil. You can see the loss of parent is this, the substrate line is here in blue. And then you can remove that and you can see how the parents, the metabolites would come in and go out over time. You can see N-demethylation and demethylation here. So you get the metabolite profiles from these 0.3 micromolar incubations. And then this is supposed to be a little picture of an iPad. Since it's all web-based, you can even do it at home on your iPad if you really wanted to. The other thing I wanted to quickly discuss is the in silico side of the software. So Mass Metacyte has a in silico site of metabolism prediction calculation that we also like to incorporate into our data sets. So I'm going to show an example of how that works with midazolam here. So this is structure of midazolam. And you can see there's many options here. You can click on individual cytochrome P450s, or you can do them all. You just click on liver, and that's what this is an example of. And it'll predict the site of metabolism based on oxidative. So sorry, not phase 2, just phase 1 oxidative metabolism. And the more bright the little red rings are is where the preference or probability for metabolism to take place. And this is really based on Cruciani's paper, who helped develop the software. I'm not an expert in this, but this is how one would calculate the probability for the site of metabolism. And it is a docking mechanism. There's docking involved into the cytochrome P450 active sites, and you can actually see that for the calculations. So I have an example of that, where this is midazolam in the binding pocket of cytochrome, I think it's 3,4A. And you can see the heme group here, and you can see how it lines up with the heme, and it's showing how the calculations were done, showing that this would be the most probable site of metabolism. So this all takes minutes, or maybe 1 or 2 minutes to do. So how well does Mass Metacyte actually predict in vitro soft spots? And if this is not your area of expertise, there's many papers on this. Many people have wanted to calculate, well, how well does this actually work? So since we run this assay every week now, we have a large data set to collect from. So we just grabbed 200 compounds from human liver microsomes and hepatocytes, and we compared the Mass Metacyte side of metabolism predictions to the actual identified soft spots. So again, this is what a soft spot would be. This is this oxidation here, this +16, this light blue line, you can see it comes in really early and fast. And that was identified as this methyl group here. So this was the metasite site of metabolism, which in this case gave us the same. But overall, out of the 200 compounds, right, how often is the top predicted site of metabolism really the soft spot? And the answer isn't very impressive. It's a coin toss. It's literally about 50%. So that was a little disheartening, but it's actually in line with most of the literature on mass metasites. So you might be asking, why am I talking about this? Well, what about the top 3 sites? Because you notice they are ranked in terms of probability. How often is the top 3 predicted sites of metabolism the actual soft spot? So what we did here, we just changed the numbers. This is just hypothetical. These aren't the real calculations. You can see I just flipped 1 and 2. And in that case, it's actually pretty good. It's 90%. So 90% of the time, the top 3 predicted sites of metabolism are actually the top. And metabolism is actually found in the microsome incubation. So that's actually quite helpful, right? That's which we now were designating that as a test 0 or T0 type in silico calculations, because we worked with the medicinal chemists and they thought this was great. Because if you're doing metabolism and you run into a molecule that doesn't fragment well, you can easily see that you have metabolism just based on the mass shift. But it's not fragmenting, so you can't locate the actual site of metabolism. Sometimes, people put a whole bracket around the whole molecule. And when you provide that to the medicinal chemist, they just say, well, thanks, Mark, that's really helpful. And it just doesn't add any value. And so at least if you're in those kind of situations, this at least helps the chemist have a starting point, because their next synthesis round is next week. And they just want to know, what do I need to do for my next step? So it still does add value. All right, I'm going to quickly go over this in silico and the in vitro qual assay in an actual early discovery program. I want to show how we did some structural evolution. So in red are the in silico predictions. So I had to mask the molecule, of course. Mark's not going to let me show a molecule. So in red are the in silico predictions for metabolism, and in blue are the sites that we actually identified. And then here are some PK parameters for those molecules. So for compound 1, you have an in vivo clearance, and it's just insanely high, right? And it has a MRT is mean retention time, kind of like a half-life. And then the LLE is a ligand lipophilicity efficiency. It's a combination of the potency of the molecule as well as its lipophilicity. So you want that number to be high, the log D, and then projected dose. So if you have something with a clearance of 16,000, it's not a drug. It's a dose of 257 grams. But taking this information, they went to the next round. And we did both in silico and in vitro MetID. And you can see, again, they overlap quite well. They were able to start bringing the clearance down, the LLE up. Still too much to be an actual drug at 18 grams. The next round, you notice they dropped the in vitro actual experiment because it kept lining up. So these were just the predicted sites. They were able to drop the clearance again. So we're down to now an actual druggable dose of 110 milligrams. And you can see the LLE is coming up, so that's good. In the next round, we did MetID. We only found 1 oxidation in this area here. Again, the clearance is coming down, LLE is coming up. Now we're getting really low, 10 milligrams. And they continued on, and this was one of the final series here. And in this case, it was able to see this methyl group out here was going to be oxidized. We didn't see that, but they changed it to a chlorine, and now you have really good compound characteristics and a predicted dose of only 2 mg. So that's kind of how we use an actual example of this design make test cycle in action. So we have this new test 0 that we use when appropriate. And before we get into our experiments to help drive and make some helps in some cases, it helps the chemist what to synthesize. And then now we have our test one or our tier one in vitro intrinsic clearance and SSID. And again, the data is coming back in about 2 weeks. So we're able to impact design cycles, right? Because if the data takes a month to get back to the chemists, so they're making molecules all the time. You really want to be able to help influence that synthetic design cycle. Let me just get through this quick. This is a conclusion to the first half. Am I doing on time? I'm doing okay. So the Biotransformation Distribution Group is really uniquely positioned to help aid design. Being centralized and modality independent, it's really been a big help. Partnerships are key. So we've formed multiple partnerships with software developers, CROs, medicinal chemistry, of course, and IT. I didn't discuss that, but definitely a lot of information technology is key to getting these successful. Software is also foundational for drug metabolism, whether it's small molecule or large molecule, and we really use, again, GUBS and molecular discovery suite. And we built our metabolic stability and qual assay to really help influence those design cycles. And turnaround times is 7 business days, so it's 7 to 10 business days. And really easy to share the data with the teams across Merck. And again, I talked about integrating the cytometabolism tools in silico. And again, for this portion, I really want to thank Chris and the rest of the team who really helped drive this forward. All right, I'm going to completely change to the next subject, where I'm going to talk about obtaining rapid tissue distribution information of drugs and metabolites. And again, in that early drug discovery space using mass spectrometry-based imaging. And I would really like to acknowledge Dr. Bingming Chen and my group, who is really the project lead for all of our label-free imaging applications. So really quick, let me review the label-free mass spec imaging toolbox that we have now at Merck. This has also been a big capability build over the last 5 years or so. We've done heavy investments in mass spectrometry-based imaging. So we have two, at Merck now, we have 2 Bruker MALDI systems. We have the FTMS, or the FTICR Solarix, and that's located in New Jersey, actually. That's not in my group, but we do MALDI imaging on it. And then recently, as Chris knows, we just last year got the Bruker timsTOF Flex. And oh, for Joe, if you need a speaker in the future, Bingming can give an update on the imaging on this, but I'm not going to be discussing that today. We have a SetQuant dropletProbe, which is a tissue extraction probe. And I'm going to be discussing that mostly today, but I'll get to that. We have access to a DESI to do surface analysis using DESI. In our group, we also have laser capture microdissection. And over the last couple of years, we've been working with Dr. Julia Laskin at Purdue, and we've been using their nano DESI for multiple applications, actually. And so we're writing papers, so I won't be able to discuss that today, but papers on that will be out very shortly. So today I'm really going to focus on the SetQuant dropletProbe applications because it has a unique space or niche where you can really get data back to teams very quickly, and that's kind of the focus of my discussion today. So what is the SetQuant dropletProbe? It's really a LEAP autosampler that moves the needle over the tissue of interest, and it hovers right above it. So you calibrate it to be above the tissue section. And then what it does is you have a solvent, and it'll just drip down onto the surface of the tissue, and it's doing the extraction, and then it'll plunge up and down about 3 times. So you're not releasing a drop, you're creating a microjunction. So a solvent microjunction between the needle and the tissue, and that's how you're doing the extraction. So this is really done in conjunction with Oak Ridge National Laboratories. And then you may have seen other applications that do something similar to this. I think Advion has one. But with the Advion system, it injects it directly into the mass spec. So there's no separation. And we had an Advion, and you really just get a lot of background signal. You really need a good signal noise, and your molecule has to ionize or be a pretty good concentration, at least in my experience. People may have better experience than I did. But with the SetQuant droplet probe, it takes that extraction, that tissue extraction, and then it injects it into an HPLC or a UPLC. So you actually get separation now on your reverse phase column, or whatever kind of chromatography you want to do. And then that goes into, in our case, we had it set up on our Orbitrap Velos. But you can connect this to whatever you want. And I'll show our collaborators have it on a triple quad, or you can put this on any kind of instrument as long as it's keyed up to your HPLC. So it really adds a lot of flexibility between the chromatography and the HRMS to see what's on these surfaces or within your tissues. So some of the positives or the pros, it's quite sensitive, which I'll go over. It's connected to, it could be a high resolution mass accuracy, again, based on what instrument you have. You are adding LC separation. I'll show that it's quantitative. We've done some curves with this. You not only are you looking for the parent drug, but it's easy to identify metabolites. And there's actually no sample preparation other than the tissue sectioning. The cons, it's for imaging, and I'm doing little hand quotes, it's very low spatial resolution. So I don't know if you can really do any kind of sub-tissue type of compartment resolution on this, because the spot sizes are about 1 millimeter. So they're a little large compared to microns that you get with MALDI imaging. And you can't do thousands or tens of thousands of spots, again, like you can with MALDI. You're only going to get a couple of spots because you have to go through this whole process. You're not going to be doing 1,000 injections on the system. I mean, you could, but I don't know if I have the time to do that. So the first application we did with this is we combined it with our quantitative whole body autoradiography workflow. So we combined this SetQuant droplet probe on tissue sections that we were doing QWBI on. So with quantitative whole body autoradiography, you have a radio-labeled molecule which is dosed, and then you see how it's distributed throughout the tissues in an organism. And you get absolute quantitation because you're measuring the emission from your, whether it's tritium or C14. And it's very sensitive because you're using you're not using mass spec as your readout. But the big issue with QWBA, and back in the day, this is always the problem, is you really don't know what you're measuring. You're just measuring an emission. You don't know if that's a parent or a metabolite, or you just have no idea. So that was the whole idea of to combine these two. Can we then go in and take these same tissue sections that we did QWB on quantitative analysis and identify what's under these under these emissions? So you get very molecular specific information. It's fast, and again, but not with really any high spatial resolution. So let me go over a quick example where we needed to examine some tissue accumulation. So we did an issue-driven QWBA study, and we saw very high concentrations of something in the liver and in the intestines, and the team really wanted to know what it was. So is that just the parent, or those metabolites, or who knows? So again, the total levels were quantified, so this is fully quantitative. But we didn't really know the qualitative side, right? What was actually giving us the signal. So we used the SetQuant droplet probe for metabolite identification. So this is the extraction from the liver. In this case, we're using 1:1 acetonitrile and water, not even any acid or anything in this one, pretty straightforward. And these are the LCMS extracted ion chromatograms. So here's the parent compound. We're able to see But then quite quickly, we're able to identify multiple metabolites. So sulfated conjugate, carboxylic acid, N-dealkylation is also N-acetylated. So right away, we're getting an idea of, not only is it just parent, but there's a lot of other species that's giving us that total signal. So you may ask yourself, well, you said this was about rapid getting data quickly back to teams, and if you have any experience with QWBA, it's not that fast. It takes weeks to sometimes months to get all that data back. So I want to give an example. We're rapidly addressing distribution questions in the small molecule discovery space, again, using the SetQuant droplet probe. So the team approached us with a lead optimization compound that had really high clearance, that was exceeding hepatic blood flow, and they wanted to know what was going on. There was a lot of different hypotheses, and other PK and other analyses weren't adding up. So they wanted to know if the parent drug was being sequestered in certain tissues, and one of the big questions was it getting to the lung? So we did a whole body SetQuant droplet probe type study to test its hypothesis, to look for levels of the substrate and see if there's any other major metabolites. And we looked at the liver, kidney, lung, intestines, and muscles about 1 and 4 hours post-dose. And it was a 30 mg per kg oral dose. And so the way we do these analyses is we try to do them in triplicate when we can. So we had 3 consecutive sections. We're lined up on the sampling plate, and then Bing took these 5 spots across the same areas of the tissues. And so the data shows the average peak area from the different tissues. So this is what the data looks like for the parent. So this is the tissue section, and the different areas are labeled here. We have the liver, the kidney, the lung, the intestines, and leg muscle, and we have multiple time points. So we have the 1 and the 4 hour. And so this was an oral dose, and you can see after both 1 and 4 hour, it's primarily still in the intestines. So this molecule was not readily absorbed as well as they wanted it to be. And then the question is, was it being sequestered into any of these tissues, especially the lung? You can see the answer is no, it's not. So here's, again, you can still see the 1 and the 4 hours, but it's very, very low. And the nice thing about running these samples on an HRMS type instrument is you're not just looking at the parent, right? You're doing full scan data accumulation. And so we're able to easily do MetID studies on those same injections, so that you can see the XIC with the parent. This is the internal standard. And very easily, we're able to find 2 metabolites, M1 and M2. And then you can go back and reprocess your data and see if these metabolites, which are identified in the liver, were they in any of the other tissues without having to do any other further laboratory-based experiments. So now this is the M1, which was actually a hydrolysis product, M2, which was an oxidation. And for M1, the levels were quite low. I mean, it's hard to say, it's not quantitative, but just based on the peak area ratios. And it was pretty evenly distributed throughout the organism in the mouse. And it was a little bit in the lung, but nothing to say it was sequestered. And then for the oxidative metabolite, again, primarily in the intestine, you can see it bleeding out over time, or being eliminated over time. And it was also in the liver and the kidney, but again, nothing in the lung. But this was done quite rapidly, which we'll get to. So being able to use this type of application of the SetQuant droplet probe in this early lead optimization has been really helpful. So in this case, we're easily able to detect the lead compound within the intestine after oral dosing, and the kidney and liver, but very little in the lung. And this really helped address the question quite quickly. We're able to identify 2 metabolites in the liver and also within the kidney, intestines, and the leg muscles, with only the M1 being detected in the lung, but very low levels. But this is the key message I wanted to get across, was the turnaround time. The turnaround time for this entire experiment, including the dosing, was only 3 days. So the team had the information back within 3 days, and it really helped the team move away from the series and kill it and move on to another series. And no-goes are just as important as goes, as you know. How am I doing? Almost done. So I'm gonna, last topic here, I only have a couple more slides. Moving beyond small molecules, so I wanna discuss using the SetQuant droplet probe to detect distribution of peptide therapeutics. So I have a couple examples. I have ASTP 7041, which is a phase 2 clinical peptide for aileron, for oncology, I believe, and then melanotan-2. These are both quick experiments, so in CD1 mice in 15-minute time points, and IV dose. So I mentioned previously, I would talk about the quantitative aspects of the SetQuant droplet probe, and this is all done in conjunction or collaboration with Oak Ridge National Laboratories. So the cyclic peptide was dosed, as I discussed, IV, in a quick 15-minute time point. And the first thing we wanted to do is it quantitative? So we had a C13 label made of the peptide. And so we had an actual internal standard, and then they had different concentrations that were extracted off the surfaces. And you can see it's actually quite linear. This was all done on a triple quad, so they had a nice quantitative assay for it. So it is quantitative if you make the calibration curves and then you extract them off. The next thing that we wanted to do was see how it would compare if you did a surface extraction using the droplet probe versus if you did a laser capture microdissection. You just cut out a hole in the tissue section. So that's the comparison between these next two plots here. This is the levels of the peptide that were identified. And then these are the different organs within the section. So we have the testes, the intestines, stomach, kidney, liver, lung, and blood as you go across the x-axis. And the first thing you can see when you compare the tissue punch, they're calling it, versus the tissue extraction, is the profiles look virtually identical. So this was exciting that the relative amounts were the same. At least you're getting that same profile. That was very exciting. But as one would expect, if you're doing an extraction from a tissue punch, and you're probably going to have a little bit more volume from tissue in there, we're getting about 6 to 8 times more from the extraction, but it's still very detectable using the droplet probe or the tissue extraction method. And then finally, we're always asked, but each tissue is going to have its own separate extraction efficiency, right, based on the different proteins and protein binding and so forth. And for this compound, I can't say this would work for all compounds, but for this compound, the extraction efficiency was about the same across all these different tissues using this extraction solvent, water acetonitrile with a little bit of formic acid. So that was also very encouraging. I'm going to quickly go over the melanotan-2 example, where this was also dosed at 10 mg per kg IV, quick time point, 15 minutes. You can see we can do this really quick chromatography using HRMS and UPLC. So the total runtime is only about, I think it's 5 minutes, 5 to 10 minutes. You can see the peak easily coming out about 3 minutes. And the nice thing about melanotan, or not nice, what's well known about melanotan is it's very quickly deamidated in vivo. And there's whole other stories on that because there's a competitor molecule for this, which is the deaminated metabolite, which is also a drug. There's a lot of literature on that if anyone's interested. But you can quickly see by using our Orbitrap or any HRMS type system, you can see there's melanotan at 512, and then this is the deamidated metabolite here at the 2+ charge state, but so 1 Dalton higher. So very easily resolved. And when you're doing these studies, so this is the tissue section, these are all the different tissues and organs within the tissue in this section. You can see the peptide was quickly brought into the liver. You can see that both the parent and the deaminated metabolite. And this is well known with peptide therapeutics. They're very rapidly renally eliminated and cleared through glomerular filtration, especially a small peptide like this. And you can see how much is in the kidney. And then there's in the other tissues as well, but low amounts. So again, this is very rapid, and it is also something that we like to apply to other programs beyond just small molecules. Last slide, this kind of the future. So Bing Ming came up to me and said, Mark, there's a lot more stuff in there than just, melanodan. And she showed me an example when she extracted from the heart region, which is primarily blood. This is how we sample blood. We sample the heart region. A lot of proteins were coming out. So she quickly did a top-down analysis. She did a re-injection and isolated this plus 16 charge state, did a top-down analysis, and was quickly able to identify it as the hemoglobin subunit beta 1. So just something that we're thinking about in the future. There's a lot of biomarkers that we could also investigate with this type of technique. All right, so with that, tonight I've discussed implementing a quantitative and qualitative metabolic stability and metabolism assay to identify soft spots and accelerate chemical optimization. And then I wrapped it up giving examples of how we're using our SetQuant dropletProbe for label-free mass spec imaging to really help where we can address issues of small molecules and peptides in the discovery space. And so with that, I would like to acknowledge all of my colleagues at work. I'm very fortunate to be able to work with such a talented team. And as I promised, I have a picture of Joe here in the middle. Again, I really want to acknowledge my entire team, our collaborators at Molecular Discoveries and Oak Ridge National Laboratories. And I would really like to thank you for your time. Thank you.
Thank you, Mark, for that wonderful presentation. We'll open for questions. Any questions?
Hey, Mark, John Masucci, I have a question for you. When you were doing your comparison, determining your percent efficiency of extraction with the microdrop with the droplet, did you choose an area to the thin tissue punch? Did you choose an area that was the same size, or did you actually weigh? Did you estimate the weight of the punch? How did you do it?
It was about the same size as the extraction. So about 1 millimeter diameter, and they used the laser capture microsection to take out that.
Okay, thank you.
And it was on a serial. There were 2 serial sections.
Did you ever try doing for instance, comparing it to homogenization of liver tissue, which is usually very uniformly distributed, and compare that to what you were getting on the droplet?
Yeah, it's definitely a little bit lower. It's not going to give you the same exact quantitative amount. So when we do this methodology, we say it's quantitative, but it might probably going to be a little bit more relatively quantitative. But I'm more interested in these kind of curves.
You want the trends, right? I understand.
I want the trends is what I'm a little bit more interested in.
Yeah. Okay. Very good. Thank you.
And you're right. When we need true quantitation in tissue, we do old school method.
Yeah, I mean, we did this kind of work in the past. We called it a microsampling technique, where we were just going in and punching out with a little syringe needle, sharpened syringe needle, and we did these extractions. And yeah, it actually compares pretty well to what you get when if you're homogenizing bulk tissue and comparing results. So it is pretty good. I didn't think you'd be able to get so high extraction efficiency. 10% is actually 10 or 20% is actually not too bad. Because the sensitivity of instruments these days, and especially since you're doing an IV dosing here, so chances are the levels are reasonably high. So that's pretty good. Yeah, looks good. Well, thank you.
Yeah, thank you.
Yeah, I have a question regarding the dropletProbe MS. What is the sampling rate of your analysis? How often do you sample the tissue?
The sampling rate's as fast as your entire LCMS cycle.
Yeah, so that's another leading question that I had is what if you can replace the LC without having to go through a column? If you can do probably something like flow injection, right? Without having any separations, what would you expect? Would that be compatible with how you would sample? I mean, is the sampling rate the rate limiting step in those instances? I just want to know, like, if you were to replace the LC and have it directly infused, is there some limitation there?
Yeah, there are limitations. So I feel the advantage, so this is the whole pros and cons. I think the advantage is the HPLC or the UPLC. I think the advantage is because when you're doing these tissue extractions, you have everything under the sun in there. And like we have done the extraction to a direct injection, and it's primarily you're just seeing background molecules. The background is so high. It's very hard, especially when you get to lower levels of your actual analyte, to pull it out. It's hard to get that signal to noise. So I think that's where the disadvantage is. The speed is definitely there. And Advian has a whole platform to do that.
Yeah. When you say speed, I mean, how many times, how many shots per second? I mean, is there
for the Advian type method?
No, in this.
So in this case, I mean, you're doing one spot every 10 to 15 minutes.
10 to 15 minutes. Okay,
that's what I mean. It's slow. That's why I have this here.
No, I was thinking whether you could do high throughput top-down mass spectrometry.
Yeah, you could.
Yeah, you showed a nice spectrum of hemoglobin, right? And you attempted to do top-down, right? And has anybody or have you seen this been used to do protein imaging?
Yes. So people, Caprioli's lab at Vanderbilt has one, and so we've been using their methods. And again, you should invite Bing Ming in the future to this discussion. She's doing tissue. She's doing trips. She's laying trypsin down, following a lot of the methodology that Caprioli has done in his lab. And then you do whole. I took that out of the. Actually, I had it in this discussion, but I took it out because I didn't have enough time. And so you can do proteomics, trypsin-based proteomics from all these tissue sections, and she does it on all different tissues. What we found is it definitely works. It's not super sensitive. You don't go too far down in the proteome. You're getting a lot of the high-level proteins, but it definitely works. And Bing Ming has a lot of good data on that.
I know you didn't show this, but what is the advantage of also doing nano-DESI? I mean, it's not as sensitive as MALDI imaging, and maybe have other problems in terms of spatial resolution, right, as well.
Yeah, no, we've
behind using nano-DESI.
Yeah, the nano-DESI, again, it's working with an academic collaborator on investigating new technologies and seeing what the advantages and disadvantages of that technology are. I'll stick to the advantages. We were able to identify, I won't go because we're publishing this now, we're able to identify some metabolites that are quite labile that we couldn't do with other methods, that we could have do with nano-DESI, and we were imaging the kidney and other tissues, and we're getting really good spatial distribution and seeing how the metabolites, different types of drug metabolites are distributed all throughout different tissues and within the tissue, so in multiple tissues. So there was some advantages there. We did a head-to-head MALDI versus nano-DESI, and the MALDI was still just a little too energetic, and we could put a lot of method development into that, where we were dissociating some of these labile phase 2 type metabolites, where the nano-DESI was able to pick it up. So that's the paper that we're working on now.
So this is with Julia Laskin or Graham Cook?
Yes, Julia Laskin.
Okay, thank you.
Mm-hmm.
Any other questions for Mark? Well, if not, I guess since we are a little bit over time, I guess we will like to thank Mark once again for a wonderful talk and a nice discussion. And thank you again for
Yes.
being here today with the Delaware Valley Mass Spec discussion group and sharing what you're doing at Merck. We really appreciate your time.
Yeah. Again, thank you. I really appreciated that. Thanks for the invitation.
Okay.
Right.
Yes. Thanks, everybody, and hope to see you guys all in about a month's time, where we will have a second virtual vendor night meeting, which will be somewhere around the second week of May. So hope you can all join us at that time. So see you.
Thank you.
Thanks, Mark, and thanks, everyone.
Thanks, Chris. Bye.
Bye.